The first step in both mitosis and meiosis is copying the DNA. This is called DNA replication. This little GIF here is actually an animation of our best understanding of how this process occurs. And you may have spent some time learning about all the different enzymes. So you have DNA polymerase and topoisomerase and helicase and all these enzymes that are required. It's a very complicated process. I think this is amazing. And this is really, really slowed down. It actually happens much, much faster than this. Now, for this class, we don't need to know all of these details, all the enzymes and what their job is, okay? But what I do want you to know is as the DNA is copied, even though it's a very, very high fidelity process, so it's very good and rarely makes mistakes for most organisms, that sometimes there are still errors. And we call an error a mistake in DNA replication. We call that a mutation. Now, when I say mutation, most people think it's, some bad thing. I get one of two responses when I say, word association with mutation. One is, some really bad, awful thing that happens to an organism that means it's not going to survive or it's going to do poorly. The other one is X-Men, right? We get a mutation and you have some superpower. Well, both of those are really not very accurate, although we can have good things that occur from a mutation and we can have bad things. So when I say mutation, in your head, hopefully you just think of it as something new, and we don't know if it's good, we don't know if it's bad. Now, when the DNA is being replicated, the polymerase enzyme has a proofreading mechanism, so that reduces the number of mistakes that are made. Not only that, but if the polymerase makes a mistake, there actually are repair mechanisms where due to some chemical difference between the new strand of DNA and the old strand of DNA, that mistake, that mismatch between the base pair can be recognized and fixed. And then, of course, there's DNA damage repair. And there are many things, chemicals, UV exposure, that can damage DNA. And if so, there are mechanisms that help to repair that. Now, for DNA replication, we're primarily interested in these first two. In the long run, all we really want to know is how often a mistake is made. So we have a DNA strand, we make a copy of it, and sometimes, rarely, but sometimes we make a mistake. And the little enzymes that are copying it, in this case, we put it a G instead of an A. Now, a little bit of a note about when we see a string of letters like this representing molecules in the DNA. By default, we only show one of the strands. We could put the other match. So A across here would be a T, T would be an A, and we could put both strands, but it's redundant because we know if we have an A on one side that we always have a T on the other. So here's the strand of DNA. After we copy it, we've made a mistake, and this base pair is now a G. On the other side would be a C, whereas before it was an A and a T pair. Okay, but we only show one strand. So just so you know. So this would represent, I've highlighted it here in red, a mutation during DNA replication. And this would be one that the proofreading mechanisms didn't catch, the mismatch repair didn't fix and catch after that. And so it's a difference. And so this is something unique. And so when we were talking about whether or not you were different from your sibling, we actually need to not only add in the variety and diversity that can be created during random assortment, the variety and diversity that can be created because of crossing over, but then also the fact that occasionally mistakes are made, new things are put in. And when we look at this, the mutation rates vary widely. Some organisms have a fairly low mutation rate, 1 in a billion. And this means that there are very, very few. But the human genome is 3.2 billion base pairs long. So even at a very, very low mutation rate, there's still going to be 2 or 3 brand new features in a human genome as it's being copied. And this would just be one small base pair difference. Okay? Some organisms have much higher mutation rates, as much as 1 in 1,000, sometimes even a little bit more than that. And so they're going to have much, much more diversity. The coronavirus and many viruses have fairly high mutation rates. Now, a mutation rate is an essential feature of all organisms. We need to have mutation. Remember, mutation is the only source for diversity. If DNA was copied 100% exactly, with 100% fidelity every single time, there would be no new diversity in organisms, and eventually, through the processes of natural selection and genetic drift, we would lose diversity in organisms, and that's non-diverse organisms just do not do well long term. So mutation is necessary, but the mutation rate is going to vary between organisms. Some organisms are adapted to a very high mutation rate, which means there's lots and lots of diversity. The downside of that means that you're going to probably have more mortality. A lot of those mutations, some of the mutations are bad, and so higher mutation rate means more mortality. Whereas individuals with low mutation rates have less diversity, but then they also have less mortality due to those mutation rates. Okay, so just keep in mind, mutation is necessary, but mutation rate varies. Some organisms are adapted to high mutation rates and some to low mutation rates. So we can kind of begin to get an idea quantitatively about mutation rates. I've made this pie chart. Now, I don't have data to back up these numbers, but I stand by them. I think they're good. I don't know that anyone has ever put data on them, and I'm giving you ranges here anyway, so I'm not going to give you exact numbers. But of all of the mutations that occur when DNA is being replicated, we can look at them and put them into one of three categories. So 3 possible outcomes to a new mutation. It can be a positive thing, right? A benefit to the organism, meaning the organism carries something brand new, and because of that, it does better than it would have otherwise. Of course, it could be negative. It could be a bad thing. Now, just a quick word as we're thinking about a positive and a negative mutation. For evolution, we really don't care about mutations that occur during the mitosis in sexually reproducing organisms. If you have a skin cell that makes a copy and it gets a mutation that's a bad thing, say cancer mutation, which happens, UV exposure, chemical exposure, other things can elevate mutation rates, and they don't cause bad mutations, but they just cause mutations. And so every once in a while, one of those mutations is going to be negative, and those cells will develop cancer. Now, that's a big impact for the individual that carries that new mutation, but it can't be passed on to offspring, so it doesn't have an impact long-term on the population. So again, for evolution, we're only interested in mutations that can, diversity and variety, that can be passed down to the next generation. So really, with sexually reproducing organisms, we're just looking at mistakes that occur during meiosis. And then finally, the third possible outcome is neutrality. A mutation can be neutral, meaning, yeah, it's there, we can measure it in the DNA, it's real diversity and variety, but it doesn't have a phenotypic impact on the organisms. So let's take a look at general numbers. And again, these are, I've made up essentially these numbers, but they're based on a qualitative understanding, and I think it's helpful to put numbers on them, but these are going to be a little bit vague. So less than 0.1%, maybe far less, but less than 0.1% of all mutations are beneficial. Less than 5% of all mutations are detrimental or negative. And more than 95% of all mutations are neutral. Now, the why we'll get to later. So I've posed the question here, but put a little asterisk by it or a little bit of a sticky note in your head, right? Why are the vast majority of mutations neutral? You might already have some ideas. Start thinking about it. Think of why this might be. But a good way to think of it, if you have a random mutation that occurs, most likely it's neutral. It's there. We can measure it when we sequence the DNA, but it doesn't impact the organisms. And so remember when we were talking about diversity of the human population and how most of the diversity that we see is present in African populations? Much of that diversity is there. We measure it, but it doesn't have an impact on the phenotype of those individuals. Of the rest, most of the remaining mutations are negative, and a very, very tiny percentage are positive. Okay, so just keep that in mind, and we'll come to the why later. So here are our base pairs. Here are the different changes that can occur. We are now going to begin to classify the different types of mutations that can occur. So you need to know these. You're going to need to review them and memorize them and go over them. We have a couple major categories and then some subcategories within those major categories. So the first major category we're going to talk about is point mutations. And both substitutions and insertions, deletions are point mutations. A point mutation, and let's add this. You can put these notes, write them down. I purposely will often make my PowerPoints a little bit of an outline without putting all of the detailed information because I'm hoping you're writing things down as I talk. You're taking notes. The very act of writing them down will help you remember it better. So point mutation here, you should write down is a single nucleotide, you could say base pair, but a single nucleotide change. Okay, that's a point mutation. And we have two main categories here that we're going to talk about, a substitution and an insertion deletion. Now within substitution, we have two main categories and then two more subcategories. A synonymous mutation is also called a silent mutation. These are mutations that have no impact at all on the phenotype. Now, let me give you an example of a synonymous mutation. We talked about it earlier. So if you remember, if you look back at the genetic code, CCC, cytosine, cytosine, those all code for a, those three in a row code for proline, which is an amino acid. And this is weird because nucleic acids and amino acids have very similar sounding names. But cytosine, CCC, all three in a row codes for the amino acid proline. You can look at the genetic chart to confirm that. And if I mutate that third position to an A, it still codes for proline. So that's going to have no impact on the phenotype of the organism because it still has the same amino acid in that gene. So that would be known as a synonymous or a silent mutation. Okay? We can also have silent mutations in non-coding DNA, but for now, we'll treat these two words as equivalent. We could also have non-synonymous mutations. So in fact, I don't have a genetic code here. I'm going to open up our old one, our last discussion, and let's actually look at the genetic code. In fact, it's probably easier if I just cut and paste it into this PowerPoint. Okay. So let's make this large. We'll get rid of it here in a little bit. So CCC proline, I mutate it to CCA, still proline. That's a substitution, but it's a synonymous or a silent mutation because it didn't impact the phenotype of the organism. But what if instead the mutation had happened at that second position? So we start with CCC, proline, but now we have a CAC, which is a histidine. So now I have a different amino acid at that codon, okay? So we call that a nonsense mutation, a non-synonymous mutation. And they come in two varieties. The one that we just talked about is a missense. So if we change one position in the codon and it changes the amino acid, that is a missense mutation. A nonsense mutation is also changing one position in the amino acid, but if it results in a stop codon. So one amino acid changing to another amino acid, it's non-synonymous because we changed the protein, but that one amino acid change is a missense. If we change an amino acid to a stop codon, it's a nonsense mutation. Now at the end, we're going to give you some general ideas about the average phenotypic impact of these different types of mutations. Okay, but before we do that, let's look at the last type of point mutation, which is an insertion or a deletion, and we treat them both equally because they both have the same phenotypic impact. In an insertion deletion mutation, we end up with a extra nucleic acid in the sequence, or we're missing a nucleic acid. And sometimes these are also called frame shift mutations. I have that word down there below number 2, because not only do they impact the codon where they occurred, but they impact everything downstream. Now, to illustrate these different types of mutations, I'm going to use an analogy here. So I'm going to bring up Word. And I'm going to write a sentence here. Now, I'm not going to put any spaces. Sorry, I'm having trouble typing here too. Okay, so again, not, Shakespeare here. This is not a really great sentence, but it makes grammatical sense and we look at it, and our brains are trained to break apart English, and so we could probably read this even without the spaces and the capitalization, the period, and everything else that we usually put as guidelines in the English language. Now, I've got more than 4 letters here. In the genetic language, we only have those 4 possibilities. But I've made every word 3 letters, so we're kind of similar to the genetic language. So a simple substitution mutation would look like this. Oops, capital. The cat ran far and the dog did too. No, this makes less sense. In the genetic language, every word has a meaning. That's not true of every 3-letter combination in the English language, but this is a substitution. But notice I only affected that one word. So if I have a change, right, I've only impacted that codon. I haven't impacted the other ones. Okay, let's go back to where we were before. However, a substitution, a insertion deletion is going to be a little bit of a different thing. So if I do that, we look at it and we'd say the cat ran far, and our brains are kind of good at picking these things out. But in the genetic language, we don't have a reader there trying to make sense of it. You only have the molecular reactions. And so this would still be read as the cat, and everything would be fine up until that point. But at that point, it would read it in every 3-letter combination. So I'm going to put spaces here. Now notice that not only did I impact the codon where the mutation occurred, right, but I also impacted everything downstream. And that's going to be important here, okay? So that would be an insertion deletion. All right, so on to our PowerPoint. Now, in addition to these point mutations, which are by far the most common type of mutation that occurs during DNA replication, the vast majority of mutations are these single insertions, deletions, or substitutions. But occasionally, there are bigger mutations. And so we're going to only look at 4 types. There are subcategories and even a few other types, but these are the 4 you need to know. And they're illustrated there on the right. A duplication mutation is a lot like a insertion, but it's over a big piece of DNA rather than just one nucleotide. So here we've got a chromosome with different genes highlighted. Notice that after we copy the chromosome, we have some of the genes represented more than they were before. They have 2 copies. A deletion is the same thing as a deletion at a single nucleic acid, but here we're missing genes. So big pieces of DNA coming in, getting copied twice, or big pieces being lost. That's a duplication and a deletion mutation. An inversion is where we flip around the genes. They were there before, they're still there, but they're in a different order. And then finally, a translocation is where we move a big piece of DNA to an entirely different chromosome. Now, let's talk before we move on about what type of phenotypic impact mutations have. And I'm going to go back here. Now, by definition, a synonymous or a silent mutation is neutral. It has no impact on phenotype. That's what it means. We still have the exact same amino acid. Now, this is still genetic diversity. It's still coded in the genes of these organisms, but it doesn't impact their phenotype. A non-synonymous mutation has a wide range of phenotypic impacts. Some missense mutations can actually be neutral. If I change a leucine to an isoleucine, which is a very chemically similar amino acid, it's very possible that we had no impact at all on phenotype. Okay, so non-synonymous mutations can range from 0 impact, completely neutral, all the way to a little bit of impact, all the way to a massive impact, completely changing the phenotype of the organism. And again, most of the time when the phenotype is impacted, right, 95% of the time they're going to be neutral. If they're not neutral, the vast majority of those mutations are going to be detrimental or negative. So non-synonymous mutations run the gamut from completely neutral all the way to big impact on phenotype. Insertion deletions tend to have a big impact on the phenotype of that gene. Now, if the gene's very important, then that's going to have a massive impact on the organism. But if it's not a really critical gene, then it may not be that much of a detrimental impact to the organism, maybe only slightly detrimental. But because insertion deletion mutations change the reading frame, they're going to have a big impact on the phenotype of the allele in which they occur. And these big mutations also often have very massive impacts on phenotypes. So larger mutations still can have a range of impacts. Sometimes an inversion or a translocation mutation doesn't impact the phenotype at all because the organisms still have all of the genes they need. They're just in a different arrangement. But most of the time, these mutations have big, big phenotypic consequences. So just know that in general. So what effect does a mutation have on a phenotype? Go back and review those and just know that, they can range from nothing all the way to very, very severe. Now, other than that, mutations are effectively random. So when a mistake occurs, there's nothing that makes certain mistakes more common than others in general. There are some very specific applications, but that's more detailed and more advanced than we need to know. So for our purposes, we're going to treat all mutations as simply random mistakes. So at the rate of 1 out of a billion, there are going to be 3 random point mutations every time the DNA is copied. So not only do we have all the diversity that can be created by random assortment and crossing over, we also have a little bit of new diversity that's brought in every time organism reproduces. And this again creates massive amounts of diversity. But effectively they are random. And this is a common misconception. I want to address it now. That if you are in an environment, let's say you're in a very hot environment, there are certain mutations that are going to allow you to survive and do better in that hot environment. But there is nothing about the environment that causes those specific mutations to occur. Random mutations just occur. Sometimes you get a mutation that makes you survive better in the cold. And if you're living in a hot environment, that's not good, and natural selection will remove that from the population. But if you're living in an environment that's hot, when the random mutation occurs, it may take thousands upon thousands of generations, but when it does occur, suddenly that organism has a benefit, natural selection can take over, and that mutation becomes more and more and more common. Okay? And so as we mentioned earlier, mutation rates vary widely, and organisms are adapted to have different mutation rates. Viruses have amongst the highest mutation rate, and that makes lots and lots of diversity, but also makes lots of viruses that are not going to be functional. But that trade-off is something that viruses is good for a virus, whereas humans and most other complex organisms have much lower mutation rates. Now, I don't expect you to memorize these graphs. It's just an illustration of that principle, and that principle you should know is that mutation is a necessary part of all living organisms. It's what creates the diversity, but different organisms have different mutation rates.